Gliomas are a diverse group of primary brain tumors originating from neuroglial cells and located mainly in the white matter. The white matter is composed of axons and lots of oligodendrocytes and some astrocytes with end feet surrounding the blood vessels extracting glucose, being part of the blood-brain barrier and encapsulating the synapse. The glial tumors are named after the cell they resemble the most and which they are thought to be derived from. Glial tumors can be derived from astrocytes, oligodendrocytes or their precursors. And you might remember when we discussed infections in HIV and HIV dementia that microglial cells originate from the yolk sac and are hematopoietic cells, and there are no glial tumors originating primarily from microglial cells, but microglial cells do have an important contribution in the spread and progression of high-grade glial tumors such as glioblastoma. And the classification of glial tumors based on the cell which they resemble was done the first time in 1926 by Bailey and Cushing, and they made this kind of pedigree with all the neural cells and their precursors. So there are neurons giving rise to neuronal tumors. There's a group of glial tumors here, and we learned a lot in the last century. So the choroid epithelium is derived from the ependym. So the plexus papilloma is also kind of a glial tumor. And we also learned that there's no such thing as a medulloblast, but we cannot get rid of the term medulloblastoma because then all the literature from the last century is useless. Gliomas can grow very diffuse and infiltrate the white matter, following white matter tracts or the corpus callosum, or they can be well defined, such as the pilocytic astrocytoma, which is a grade 1 tumor, and the ependymoma, which is derived from the radial glial cell. The pilocytic astrocytoma is named after the bipolar, elongated, hair-like shape of the astrocytes. The high-grade tumors that infiltrate the brain can either spread along the blood vessels, so perivascular as you see here, or along the white matter tracts, having the same direction and shape as the axon. And you can see, for example, often spread along the corpus callosum, arcuate vesicle, or uncinate vesicle of these high-grade tumors. Initially, it was thought that glioblastomas arise mainly from neural stem cells, but there was an article in Science 2012 where they injected a lentivector in mice and they mutated NF1 and P53, and this combination is often seen in glioblastomas. And it was shown that also astrocytes and oligodendrocyte precursor cells can give rise to gliomas. An article from the late 90s in Pathology nicely illustrates that the pilocytic astrocytomas have a totally different genetic profile than the grade 2, 3, and 4 astrocytomas, and that primary glioblastomas have a different genetic profile than the secondary glioblastomas. And although the first WHO classification was based solely on histology, in 2016 the molecular information was added, and this molecular and genetic information gives some guidance for the prognosis and therapeutic options. And in the most recent classification from 2021, things were restructured and tried to be simplified, and the genetic information was even more important. So, although on imaging we cannot predict the exact genetic profile, there are some clues, and we are going to discuss the different glial tumors in the next vlogs and see if there are some clues for the different tumor types. And we will start with a pilocytic astrocytoma.